A remarkable new study, just published in Science Advances, has delivered some of the strongest evidence yet that quantum mechanical processes are directly shaping the molecular machinery inside your cells. The study, led by Zede Haghigi, Siguenza, and Craddock at the University of Waterloo, along with Christoph Simon at the University of Calgary, focuses on microtubules, the protein polymers that form the structural backbone of every eukaryotic cell. Microtubules are essential for cell division, intracellular transport, and neuronal growth, and their assembly depends on a chemical reaction, GTP hydrolysis, that requires magnesium ions. Here's where it gets interesting. Magnesium comes in three naturally occurring isotopes: magnesium 24, magnesium 25, and magnesium 26. Chemically, they're virtually identical. Same bonds, same reactions, but magnesium 25 has one unique property: it carries a non zero nuclear spin. The other two have a nuclear spin of zero. Now, classical biochemistry says that shouldn't matter, but quantum mechanics, specifically something called the radical pair mechanism, says it absolutely can. The radical pair mechanism describes how pairs of molecules with unpaired electrons can oscillate between two quantum spin states, singlet and triplet, and those different states lead to different chemical outcomes. The rate of that oscillation depends on nearby nuclear spins and on external magnetic fields, even very weak ones. So, the researchers tested this directly. They ran microtubule polymerization assays using each of the three magnesium isotopes, both in Earth's normal geomagnetic field and with a weak 3 millitesla magnetic field applied using a Helmholtz coil. They controlled for temperature, pH, magnetic field uniformity, and paramagnetic impurities, all the confounding factors that have made this kind of experiment notoriously difficult to reproduce. The results were unambiguous. When the 3 millitesla field was applied, magnesium 25, the isotope with nuclear spin produced a dramatic increase in microtubule polymerization. The other two isotopes showed no response to the field at all. The statistical significance was extraordinary, with a p-value over the 6 sigma level, meaning there is a 1 in 10 million chance the results were from random chance alignment. This rules out classical explanations. It's not a mass effect because magnesium 25 sits between the other two in mass. It's not temperature. It's not field inhomogeneity. The only variable that explains the result is the quantum spin property of the magnesium 25 nucleus, interacting with radical pair electrons through hyperfine coupling, amplified by the applied magnetic field. The team also built a theoretical radical pair model that achieves quantitative agreement with the experimental data and makes testable predictions for future experiments at higher and lower field strengths. What makes this so significant is the biological context. We're not talking about an exotic sensory system like bird navigation. We're talking about microtubule assembly, one of the most fundamental processes in cell biology. This suggests that quantum spin dynamics may be woven into the basic operating logic of the cell. From the perspective of our research program, this is deeply resonant. We've been investigating microtubules as more than passive structural elements, as ordered lattices capable of supporting exciton transport and coupling to electromagnetic fields. This new study adds a critical piece. Quantum effects aren't just acting on the cytoskeleton, they're integral to how it's built. Check out the full article below.